as we finished our last lesson, we started talking about how there's mutations, and a mutation just means it's a change in genetic code. Like when you make a protein, when you have the different codons put together, the different letters, if there's a change at all, it's going to be called a mutation. Do you guys know of any kind of mutations where there's a change in the genetic code? Okay, so yeah, sometimes ultraviolet light can change your genetic code, right? You can have your cells damaged by ultraviolet light. A tan doesn't necessarily change your genetic code, but too much tanning can. Right, it can mutate that cell. Okay, so is a mutation a good thing or a bad thing? Depends. So there are some good ones, there are some bad ones, and that's kind of what we're looking at today. Is we want to look at some different mutations, and we want to decide if mutations are a good thing and if mutations are a bad thing, or sometimes they don't even matter. It's kind of the point of what we're doing today. So I have some different mutations we're going to read about, and we're going to decide how they affect the organism and whether that's good or whether that's bad. Before we do that, I have a video. I'm going to have to switch over to presenting a tab so you get some audio. All right, so if you don't get audio, let me know.
I'm a bit of a perfectionist, but a bit of a messy, unable to decorate my classroom kind of perfectionist. Actually, maybe I'm not. I wanted to make my classroom inviting and exciting, and I really wanted my students to walk in and think, this is amazing. I could never make it look like what I imagined of, but the most amazing thing did happen in the week to follow. I knew a colleague who had a friend who had a son who was going to college. Confusing. But anyway, that son could not take his pet snake to college with him. Some rolled outdoors with snakes. My colleague asked, would you like it? They said they can give it a cage and everything. It could be a classroom pet. I didn't even have to think about it, of course. So Spike, a Texas rat snake, became a classroom pet. An amazing, popular one at that. He was friendly, well, for a snake. He'd let you hold him and not bite. That year I was the only one in the science wing with a pet snake, and I'm pretty sure there were some people that wondered, what was the point? Well, I'm big on relevance, so anytime I would teach a biology concept, I found some way to work in Spike. Predation? Well, let's talk about Spike's appetite for rats. Mitosis? Let's talk about why Spike even needs to make more cells. One day in tutorials, a student asked me, since Spike's parents were bred in captivity, did you ever see Spike's parents? Do you think Spike's parents looked like him? You can't ask a question like that and not expect an answer. It was a great question because we were getting close to our heredity unit. Heredity is about how traits are passed down from parent to offspring. We've made a playlist of our videos that focus on heredity, including reproduction, how to track inheritance and pedigrees, how to solve genetic problems in Punnett squares, and understanding different Mendelian and non-Mendelian inheritance. But you really can't delve into those and study heredity without understanding DNA, chromosomes, genes, and traits. And that's what this introductory video is going to focus on. So back to the student question. Spike has traits. Patterns on his body, his size, these traits are coded for in his DNA. Some of the traits he inherited can be influenced by the environment. For example, if Spike had not had the nourishment he needed, that could affect his size. DNA is not just one big code buried deep down in an organism like some treasure. Spike's DNA is in the nuclei of nearly all of his body cells. He inherited his DNA from his mother and father. I can't know for sure what Spike's parents look like, but I do know that Spike inherited his DNA from them. One fun fact, many snake species can reproduce asexually. Had that been the case for Spike, he would have inherited all his DNA from only one parent. But it would still be DNA coding for traits. Just like Spike, your DNA codes for your traits, and your cells can't function without it. DNA determines how tall you are, what color your eyes are, what color your hair is, or even if you're at risk for certain diseases. Also, like Spike, your entire DNA code is in most of your body cells. That's why in those crime-solving shows, which we may or may not absolutely love, a criminal can sometimes be caught by just leaving a cell from a hair follicle behind. DNA has a beautiful structure, and that structure will help you understand how inheritance works. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid. If you remember from our biomolecules video, nucleic acids are a type of biomolecule. Nucleic acids are made up of building blocks called nucleotides. Nucleotides have 3 parts. One of them is a sugar called deoxyribose. One of them is a phosphate. We sometimes say DNA has a sugar-phosphate backbone. But the most important part of the nucleotide is the base, because the sequence of the bases can actually code for traits. So as far as the bases go, there are 4 types of bases in DNA. A lot of times they'll just use the letters A, T, C, G. The A is for adenine, the T is for thymine, the C is for cytosine, and the G is for guanine. These bases actually pair in a specific way, and there's a popular mnemonic that can help you remember which of them pair together. Apples in the tree, that tells you the A for apples, T for tree, because the bases A and T go together. The other verse is car in the garage, and that can help you remember the base C always goes with the base G. Regardless of whether we're talking about spike, or a plant, or a protist, or a human like you, these are the DNA bases in living organisms. But the amount of DNA bases overall, and the sequence of those bases we mentioned, will vary among different species, and also among different individuals. But it's likely to infer that Spike has a sequence of DNA bases that is more similar to his parents than he would to, say, rattlesnake. DNA has 2 strands, so there are nucleotides running up one side, and there's nucleotides running up the other side. The bases are what pair in the middle. The bases are held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. Portions of DNA make up genes. We can say, for example, that this part of the DNA here makes up a gene. Genes can code for proteins. Proteins have a huge role in expressing a trait. For example, let's consider your own eye color. Human eye color is actually pretty complex. It's determined by many genes. The genes can code for proteins involved in producing the eye color pigment. But proteins coded for by genes, they can play a wide variety of roles besides just your eye color. Proteins are involved in transport, in structure, in acting as enzymes that can make all kinds of materials, in protecting the body, and so much more. We do want to mention that not all genes are used to make protein, and there are parts of DNA that are non-coding. And even though nearly all of your body cells have your entire DNA code, your body cells may only use certain portions of those genes. Genes can be turned on or turned off by a variety of mechanisms. We call that gene regulation. Check out our video on that. Now, you have a lot of DNA. When it's compacted, it can be organized into a unit called a chromosome. Very helpful when you're trying to make more cells and you need to get the DNA into those new cells. Chromosomes in your body involve DNA wrapped around protein structures. Humans have 46 chromosomes. That means nearly every body cell in your body has 46 chromosomes. Human sperm and egg cells, on the other hand, each contain 23 chromosomes. So you receive 23 chromosomes from your mother, 23 chromosomes from your father, to give you your 46 chromosomes, your genetic code. So let's do a recap of the big picture. Here's a single chromosome. You see genes on this chromosome. These genes consist of portions of DNA. DNA is made up of nucleotides, and it's these bases here, the sequence of them, that makes the difference in coding for traits. Understanding this foundation is essential for understanding heredity, whether you're talking about you or Spike. So this may bring up more questions now, like where do dominant and recessive traits come in? What about alleles, and how do you arrange those around a Punnett square? Well, you can explore those concepts and more in our heredity playlist. Well, that's it for the Riva Sisters. We invite you to stay curious.
That's a lot of information in the video. Were there any words that you heard in there? Something that you knew or you'd already heard of from the last video that we watched? Okay, chromosomes, yeah. We know about chromosomes, and in chromosomes do you have 46 or do you have 23? Yeah, you have 46 total, because they come in pairs. You have 23 that you get from your mom, and you get 23 from your dad. But they talked about something different. They said that some snakes can reproduce asexually. What does that mean? Exactly. They don't need a mate, right? They get all the chromosomes they need from that one parent. So if you're going to reproduce asexually, you give 46 total rather than the 23. So do you think those would be similar? Like if I reproduced asexually, would the baby be more similar to me than if I had my son with my wife? What do you think so? It would be. It would be an identical copy of me. You're right, because there's no more genetics aren't coming from any place else. There's no other information coming from another source. So if you end up having asexual or cloning, the babies are just like the parents. How about another word that you heard that we had already talked about, or you had heard before that? Genes and DNA. Okay, so that's kind of a weird thing. DNA makes up genes, but a strand of DNA is made up of genes. So if I have a strand of DNA, usually like 6 feet long, there's little areas that end up being genes of that DNA. So it makes that gene up. But then it's in a long strand, so there's a bunch of different genes on it. So it kind of gets strange on a strand of DNA has genes on it, but DNA itself makes up genes. I don't know if that gets confusing for you guys or not, but so what does DNA do? What's its job? Does it go around the cell and give off energy? Does it go around the cell and move things around? No, we don't have that one. Okay, it's a set of plans, right? DNA's whole function is a set of plans. It's used to make proteins, and then the proteins go out and do the work. They can be used to make parts of the cell, like the structural parts of the cell. They've been used to do different chemical processes if you're breaking something down, or they can do that. So protein is kind of the workhorse of the cell. It's what the cell's made of and what makes the cell work. DNA is the instructions, right? It doesn't actually do the work. And there's a code. Did you see the AT and the CG? The base pairs, they call them? Is that new, or is that something we already know about? Something new? Okay, so we will talk more about that. But those base codes, that's where the mutations happen. That's where the change happens when a C, a T, or an A, or a G, a C or T, or a C or a G. Did I say A, T, C, G? Sorry. If one of those changes, that's called a mutation. So let's go ahead and take a look at some mutations. And, oops, sorry. So myostatin-related muscle hypertrophy. Have you seen those little kids? Like, you've seen them on YouTube, or you've seen pictures of them. They look like they're muscle men, or they're bodybuilders or stuff. So that's actually a mutation, right? There's something different about them that makes their body grow like that. And this is the myostatin-related muscle hypertrophy. It's caused by a mutation in the MSTN gene. A normal MSTN gene codes for the myostatin protein, which functions to limit how much muscle cells grow and divide. So your body has a regulator, has something that stops your muscles from growing. A mutation in the MST causes a loss of function, which changes the instruction of the gene enough to stop making myostatin molecules that work. These non-functioning myostatin molecules are not able to bind to the receptors on the skeletal muscle cells. Without myostatin to regulate the muscle cell growth and division, cells grow bigger and divide more than usual, leading to a boosted muscle mass and sometimes strength. This condition results in people that can look like ripped bodybuilders without doing any of the heavy lifting. So in this mutation, there's no gene, there's no protein that tells the muscles to stop growing, that they're growing too much. So we have to answer a couple questions. So in this mutation, how is the function of the protein affected by the structural change to the gene? So when it mutates, what happened to the protein? Did you need to go back up and look? So we need to find something about protein in here. Because the non-functioning myostatin molecules are not able to bind to the receptors. Right here, this sentence. Yeah. So how does that change in the structure of the protein affect the structure of the organism? So for the little kid, how is this person changed because of not being able to bind? It increases muscle mass, right? It allows the cell to grow. Awesome. And do you think it's harmful, beneficial, or neutral? Neutral? Okay. Harmful? Benefit? We got all three. Neutral, harmful, and beneficial. Somebody tell me why you think it is neutral. We probably need to do more reading to really determine whether it is neutral, harmful, or beneficial, but just based on what you see in the picture and what you read about. Okay, so it doesn't seem to cause any physical harm, and it doesn't provide any health benefits. So that would make it neutral. It doesn't really help. It doesn't really hurt. All right. Somebody said harmful. Why do you think it might be harmful? Yeah, I'm going to ask beneficial next. Okay, so it wasn't meant to be that way, so that means it's bad. Okay. And what about beneficial? Because he's stronger and healthier? Okay, I could see that. So let's go on to the next one, and maybe it'll help clarify some answers for this one when we look at a new one. Osteosclerosis, right? It's bone hardening caused by a change in the LRP5 gene. A normal LRP5 gene codes for a receptor protein. It works with another protein to send messages from the outside of the cell to the cell's nucleus. This affects the way cells and tissues develop and is important for many cellular activities. Certain mutations in the LRP5 gene can cause new proteins to be made that create new signaling pathways on bone cells, making them grow denser and bulkier. People with these conditions may be less likely to break a bone or fracture their skull. However, some people may have too much bone hardening, which can result in severe bone growth that could cause their skull to put too much pressure on their brain, or their bones to put too much pressure on their nerves. Okay, so we have the same set of questions for this one. How do you think the function of the protein affected the structural change to the gene? So not how does it affect the organism, how did it affect the protein? Didn't you guys find that in the reading? It's kind of right in the center. Yeah, they make new signaling pathways, right? So it actually changes the protein. It's a different protein. Instead of a protein that doesn't make something stop, it actually makes something new. So how does that change the structure of, how does that change the organism? Because you have these new signaling pathways, the organism has, okay, it makes the bones denser. So what do you think? Good, bad, or helpful, harmful, or neutral? Okay, we have a harmful. We're going to no beneficial on this one. Okay, so two of us are going with harmful. What do you think is harmful about that? Or what did you say? Too much growth and pressure on the brain. Yeah, that is definitely a harm. So what about neutral? Why would somebody put neutral? Right, because it doesn't affect some people. Right, for some people, they don't get that too much bone. They don't get the pressure on their skull. So some people have this mutation, and it doesn't really affect them the same way. So if I look at this and I look at the one previous, do you think this one is more harmful, or do you think it's more neutral than the one before? If you had a choice, which one would you have? Would you be the super built kid, or would you be the super strong bone kid? You could just put first or second. The first one is the one where the kid was really ripped up, and the second one is the bones. So yeah, everybody's going with first. Yeah, because you don't really have that bad thing. You don't have that harmful thing like pressure in your brain. You know, it may be kind of bad, but it's not as bad as this one. So that may help us kind of put the first one in perspective a little bit. Okay, lactose intolerance. Lactose intolerance is caused by a change in the LCT gene. This gene codes for lactase, an enzyme. Enzymes are special types of proteins that help to digest lactose, a sugar found in milk and other dairy products, and is certainly only expressed generally only expressed in infants and young children. Most adults experience a decrease in the activity of the LCT gene, and therefore a decrease in the production of lactase. A mutation in the LTC gene causes the gene to be expressed throughout adulthood, therefore causing the production of lactase beyond infancy into adulthood. Roughly 30% of the world's population exhibit lactase-persistent version of the LCT gene, allowing them to consume dairy products into adulthood without discomfort. Okay, so how did the protein change in this one? I'll go back up to the slide. So we're looking for the change in protein. Yeah, okay, so it keeps on being expressed, right? Usually it kind of tapers off, and in this case, it doesn't taper off. So how does that affect the organism? Right, that lets them drink milk. Are any of you guys lactose intolerant? Like you don't feel good after you drink milk? Do you know, are your parents lactose intolerant? Like they won't eat cheese, they won't eat milk, ice cream, and if they do, they kind of feel bad. Because usually, right at your age, you're kind of right on the edge of it, where you're going to start slowing down with your lactase. So if you can't drink milk as an adult without feeling bad or having an upset stomach, that's normal. If you can drink milk when you're an adult, that's a mutation. Kind of sounds backwards, right? You would think that if you get sick and you can't drink milk, that's the mutation. It's the exact opposite way. So normally humans are supposed to, the natural state is, you're supposed to not drink milk as an adult. It's supposed to kind of make you sick. You're supposed to be able to drink it as a kid, then it tapers off. But you're a mutant if you can drink milk. Yeah, I can drink milk too. My lactase has not tapered off, so I'm a mutant. So, do you think that's good, bad, harmful, beneficial, or neutral? That you could drink milk into adulthood. Neutral. How about back in the past? You know, maybe like 500 or 600 years ago, maybe 1,000 years ago. Do you think it was helpful or do you think it was harmful? You wouldn't know. You're not going to guess. Yeah, it probably was helpful because right now we don't usually have malnourishment in the United States. It's not a huge problem for us. We can get our vitamins and minerals, and we can get our calcium from different places, right? If you can't drink milk, you can take calcium pills that you buy at CVS or Walmart or wherever. So you can get calcium different places. You don't have to get it from milk. Before you had pharmacies and all these different kinds of pills to get your things, and malnutrition was more of a deal. It probably is helpful that you could find it. It gives you another source of vitamins and minerals. So at one point, it was probably helpful. Now, I think it's probably more. Okay, so let's look at another one. Smith-Lemli opposite syndrome is caused by a mutation in the DHCR7 gene. This gene codes for an enzyme, a special type of protein, that is involved in the production of cholesterol. Cholesterol is an essential component of the cell membrane and tissues of the brain. Mutations in this gene result in errors in the amino acids that make up the enzyme. This changes the shape of the enzyme and reduces its ability to function properly. Different mutations in the gene result in the production of enzymes with reduced function. Completely non-functional enzymes, or in some cases no enzyme at all. People who have SLOS are unable to make enough cholesterol to support normal growth and development. A person who can't make enough cholesterol will therefore experience poor growth, developmental delays, and mental retardation. In addition to mental retardation and poor growth, common physical signs of SLOS are cleft palate, microcephaly, and a polydactyl or an extra finger or toe. Okay, so that one's a little bit different. What happens to the protein? Yeah, it's shaped differently or it doesn't work at all. And how does that affect the person or the organism? Yeah, that's the poor growth, the mental retardation, developmental delays. So helpful, harmful, or neutral? There's no neutral. Anybody want to go beneficial? Yeah, so this one is obviously harmful, right? Those are pretty big problems that you're going to end up with. So if we looked at all of them together and went back and voted for harmful, neutral, and beneficial, we might change our answers, right? Because this is definitely bad. Nobody's going to argue about that. Nobody's going to say, well, right? So some of the other ones that aren't as bad as they may have appeared at first, right? Okay. So kind of the point of looking at those different ones is we want to look and see that a mutation isn't always a bad thing. Generally, if I say, oh, you're a mutant, you're not real happy about that. It's like an insult. It's not necessarily an insult. It doesn't mean it's something bad. Sometimes mutations are good. And over evolution, over the process of an organism changing over time, we keep the helpful ones, right? So we're just a whole group of mutations. As a organism, we're not the same as we were 1,000 years ago. Each generation, there's little tiny changes, and the changes that are helpful stick around, and the ones that are harmful tend to drop off. And some of the neutral ones, they just kind of stay by chance, not necessarily because you need them. So sickle cell disease is actually a mutation, right? To bring it back to our anchoring event. And what ends up happening is you have this HBB gene. And in an HBB gene, it codes differently. It codes the beta subunits, right? There's two alphas and there's two betas. The alphas hold oxygen, the betas hold oxygen. But the HBS gene will stop producing betas that can hold oxygens. So that's where you get the sickle cell. That's why they stick together. That's why they clog up the different blood vessels. It's because of this mutation. But the thing about the mutation is when you have those long strands that we looked at, and it starts to pull the cell together, right? Instead of being the nice donut shape, it starts to be the crescent shape. That stretched out the membrane on the outside of the cell. When you stretch out the membrane, the entrances and the exits of the cell tend to get bigger, and you have leakage. So that's bad. So the body attacks those. If you have sickle cell, your body is fighting your blood. Not every one of your red blood cells is going to be a sickle shape. Some of them are. So your body kind of hunts those down, and it destroys those. But there's a disease called malaria, and you get it from insects, from mosquitoes. Right? A mosquito will bite you, it'll give you malaria. And the malaria lives in the red blood cells. So if you have sickle cell anemia, your body's hunting down those red blood cells. So if you get malaria, and the malaria gets into a sickle cell, your body destroys it automatically. So it's actually helpful if you have sickle cell anemia and malaria, because your body is fighting it off better than it would without it. So sickle cell, I thought that was a bad thing. We said it was real painful, right? And it can stop oxygen flow. You get reduced oxygen. That's bad. So how could it be helpful? Right. It helps to fight off malaria. That's why we said that if you are from Central America or Northern South America, right, the tropical part of South America or the tropical part of Africa, you have a higher chance of getting sickle cell. Because at one point, people in the area that you were evolving in were dying from malaria. And the people with sickle cell were living. The people with malaria were dying. So sickle cell got passed on. And it was more helpful than it was harmful. Kind of weird. We'll go more into that idea. That's going to actually lead into our next unit. We're going to talk about evolution. So we're going to leave that there. But sometimes diseases start off as something that's helpful and turn into something that's harmful. So what we want to focus on today is we want to kind of look at how this happens. Open up your codon analogy. It's going to end up looking like this. But at the same time you have this open, you're going to have to have your dashboard open, or I'm sorry, your module open. With a Google Doc, you don't have the translator. Right? You can go here and you can open up the worksheet and you can write on it real easily. But it didn't transfer this. So you can't see this part of the code. You only get this bottom part down here. So you're going to have to look at this at the same time. So I have this open. I'll give you a second to get that open. Are you guys good? Okay, so what ends up happening is you have a translation. You open up the DNA and new pieces stick on. If the right pieces stick on, you get proteins. If the wrong, if they end up substituting something that's wrong, it makes a different protein. So what I need you guys to do, this is a code, and then one of these is mutated. So they work in pairs of 3. It's called a codon. That's why this paper is called Codon Analogy. What I want you to do is I want you to go through and I want you to highlight 3 letters at a time. So I'm going to highlight 3, and you do whatever color you want to do. And I'm going to leave 3. Then I'm going to highlight 3. Then I'm going to leave the next 3. And this will help you read it if you highlight the 3 at a time. Because sometimes it gets confusing. You start reading in the wrong place. Because this, AUG, that's going to be a code for a protein. GUG, the next 3, that's code for a different one. This is a code for a different piece, a different piece, and a different piece. So if I go through and I highlight groups of 3, it'll help me be less confused. They do that in math too, right, with the commas, to try to break up the numbers, make it easier to see for you. Group of 3, 3 blank, 3 blank. So what I need to do is I need to figure out what AUG stands for. So I'm going to go back here, and I need to find AUG. So the first letter is A, so I'm going to come to this thing, and I'm going to go down to A, and I'm trying to find AUG. So the second code is in this area, so I need to find U. So here's A, and here's U. So I'm looking right in this area right in here. It's one of these 4. And then I come over to the third area, and I find G. So A puts me in this block, U puts me in this column, and G puts me in this row. So what that stands for is start. We'll do that again because it gets confusing. So right over start, right over AUG, I'm going to put start. And that tells you to start building a protein. The next one is going to be GUG. So I'm going to go to the same place, and I find G, so it puts me down in that. And then I find U. So I'm in this block, in this column, and it's GUG. So you guys look up GCA and tell me what you think it is. So the first letter is G. That makes it this box. Second letter is C. So that's this box. And then the last letter is A. So it should be O. Right, it's G. So it puts us down here. C puts us in this column. And then A. GC. We're looking up GCA. Right, GCA. G. C. A. So that's a lowercase o. Yeah, that's why we're practicing, because it's easy to confuse. We're practicing to make sure that you kind of get it. I'm going to let you do it on your own. I'm not going to make this take forever. So the next one you need to look up is UUA. Sorry, UAA. I'm looking up UAA. Awesome, right? U is this box. A is this column. And then A is this row. So it's exclamation point stop. Oop. So that codes for the word hello, right? So it's going to start building here. And it's going to make an H, then an A, then an L, then an L, and an O, and then an exclamation point, and that's where it stops. If it doesn't have a start, it doesn't start building a protein. If it doesn't have a stop, it doesn't ever stop building a protein. So whenever I'm building something, I'm using these codes. I've got to have a start, and I've got to have a stop. Otherwise, I have a problem. So what you're going to do is you're going to come through and you're going to decode these. And then they have kind of the same questions that we were doing before. What's the effect of this mutation? And what would have changed? Then you get another message. What's the effect of it? What the effect of it, and what changed? Okay, and there's 10 questions that you're going to work through today. Okay, so go ahead and as you run into problems, let me know, and we'll talk our way through them.